Hey guys, welcome to this week's lesson from Chapter 12, Section 4, over gene regulation and mutation. We've talked a little bit about mutations in the past, and you guys definitely know what genes are and the role of genes. Genes give us all of our traits, basically. Um, Gene expression is regulated by the cell, and mutations can affect this expression. I want you guys to stop, pause the video for 1 minute, and think about mutations, what they are, what are some you know of. Think about all the things you already know about mutations. Go ahead and pause it for a minute and come back. All right, hopefully you had time to pause the video and think about that, but we're going to look at mutations here in just a second. First, our essential questions for today are: how are bacteria able to regulate their genes by two types of operons? How do eukaryotes regulate the transcription of genes? And what are the various types of mutations? If you can answer those three questions when we are done with this lesson, you know exactly the things that you need to know. Well, first, let's look at prokaryote gene regulation. So what gene regulation is, it's the ability of an organism to control which genes are transcribed in a response to the environment. We have what's called an operon, which is a section of DNA that contains the genes for the proteins needed for a specific metabolic pathway. And an operon contains several things. It has an operator, which is kind of like an on-off switch. It has a promoter, which is where RNA polymerase is going to bind. It has a regulatory gene, and it also has genes that code for very specific proteins. So there are a couple types of operons. The first one we're going to look at here is called the trip operon. Tryptophan synthesis occurs in 5 steps controlled by the trip operon. And the trip operon is a repressible operon because it's usually repressed or turned off. We can see in these pictures here, this picture represents a trip operon when it's kind of in the on position. This picture represents a trip operon when it's in the off position. We also have what's called the lac operon. When lactose is present, E. coli can synthesize an enzyme to use it as an energy source. The lac operon is an inducible operon because an inducer binds to the repressor and inactivates it, which allows transcription to be turned on. So this right here, this picture represents a lac operon in the off position. This picture represents a lac operon in the on position. Now that was for prokaryotic gene regulation. It's a little bit different with eukaryotic gene regulation. So transcription factors ensure that a gene is used at the right time and that proteins are made in the right amounts. Complexes that guide the binding of the RNA polymerase to a promoter are there, and regulatory proteins that help control the rate of transcription are there as well. So the complex structure of eukaryotic DNA also regulates transcription. So there are various things that goes into regulating our gene expression, also transcription. When we're talking about eukaryotic genes, it's way different than it is in prokaryotic organisms. So we also have these things that are called Hox genes. Hox genes, they have very specific roles. So gene regulation is crucial during development and cell differentiation. And what I mean by that is when an embryo begins to form into the organism, the cells are all the same, but eventually certain cells, think about your own self, certain cells have to become hands, and certain cells have to become feet, and certain cells have to become hair and eyeballs and skin and all the things that make up the human body. The Hox genes control cell differentiation. It's short for home box gene group. We just call them Hox genes. And again, they control cell differentiation. So they control how cells turn into the different types of cells. Hox genes are transcribed at specific times in specific places on the genome, and they control what body part will develop at a given body location. this picture here shows kind of a color code. We have this embryo of a Drosophila, which is a fruit fly. This embryo is color coded. Well, if you look at the adult Drosophila or the adult fruit fly, the same colors as the bottom shows where the genes kind of differentiate into very specific parts of the body. That's what Hox genes are, genes that control cell differentiation. Okay, we also have what's called RNA interference. RNA interference, or RNAi, can stop the mRNA from translating its message. So single-stranded small interfering RNA and protein complexes bind to mRNA and stop translation. So these things get in there and basically make translation stop. Now mutations, we're going to see that there are many types of mutations. We have, well, what a mutation is a permanent change that occurs in a cell's DNA. We have point mutations, and we have what's called insertion deletion mutations. A point mutation involves a chemical change to just one base pair. And then we have what's called missense substitutions and nonsense mutations. A missense substitution, that's when DNA codes for the wrong amino acid. So, oops, I made a mistake. A nonsense mutation, you have a codon for amino acids, and they become a stop codon. Stop codons are a code that tells RNA to stop being transcribed. It stops creating RNA, and it accidentally makes one where it shouldn't be. Then we also have insertion deletion mutations. These are additions or losses of a nucleotide to the DNA sequence, and it causes what's called a frame shift. And what I mean by a frame shift, imagine you have your codons, so A, C, C, G, G, A, like whatever. If you pull one of those letters out, the A, C, C, G, G, A, if we pull one of those G's out, and then it's A, C, C, G, A, whatever was next, the whole codon shifted. It messed up. The 3 base code is completely different now because something was either taken out or added in. So that's called an insertion deletion mutation, and it causes a frame shift. Now, protein folding can cause some changes here. So even small changes in the DNA code can cause genetic disorders. The change in one amino acid can change the sequence of the protein enough to affect both the folding and stability of the protein. So one little mistake in there can cause damage to the whole DNA sequence. Now, mutations have many causes. They can occur spontaneously. DNA polymerase can attach the wrong nucleotide, but this is super rare, and usually it corrects itself. And then certain chemicals and radiation, these are things called mutagens. They can damage DNA. So anything that can cause harm to a DNA is called a mutagen, in the sense that it can cause harm and create a mutation. Chemicals can cause mispairing of base pairs, or themselves can substitute for base pairs. And then we have high energy radiation that can eject electrons from atoms within the DNA molecule, leaving behind unstable free radicals. So chemicals and radiation are very bad and can cause mutations, which we do not want to occur. And then there is a difference between body cell mutations and sex cell mutations. Somatic mutations in our somatic cells, which is all of our cells except sex cells, these are not passed on to the next generation. So if you go out in the sun too much and it causes skin cancer, those are somatic cell mutations, and you're not going to pass that along. But mutations that occur in sex cells, those are passed on to an organism's offspring, and they will be present in the cells of the offspring. So that's the difference between somatic cells and sex cells mutations. And that is it for our lesson today. If you need anything clarified, go ahead and rewind through the video or take a peek at the PowerPoint. And when you're finished, make your way over to this week's discussion board and respond to the prompt.